In this video, we're going to talk about the biochemistry of corticosteroids. Now, corticosteroids might seem like a daunting medical subject to study. I mean, just take a look at the scheme. It feels intimidating enough to memorize. I can feel that. However, this video's got you covered. Now, let's start our lecture with the definition of corticosteroids. Corticosteroids are steroid hormones, obviously. And now you might be thinking, so what? What's so special about the steroid hormones? Here's the trick. Steroid hormones act via binding to intracellular receptors instead of binding to surface receptors. Let me clarify this with a drawing. Here's a cell, and this red rim right here is the cell membrane, and we have surface receptors present on the cell membrane. And let's suppose that this green rhomboid right here is the steroid hormone. Now, the steroid hormone is not going to bind to these surface receptors. The steroid hormone will ignore them, and instead it's going to pass through the cell membrane as if it's nothing. Why? Because the steroid hormone is a lipid, whereas the cell membrane consists of lipids too. And so the steroid hormone would be able to effortlessly diffuse across the cell membrane via passive diffusion without any help of protein channels or carriers. And once inside the cell, the steroid hormone is going to bind to an intracellular receptor waiting for it, and these two will form a complex together. And this complex is going to modify gene transcription and translation, with the end result being modification of the structure and the function of the cell as a whole. Now, some of the steroid hormones are produced by the adrenal cortex, which is a part of the adrenal gland. And so we call these steroid hormones corticosteroids. Cortex, cortico. But please bear in mind that not all steroid hormones are produced by the adrenal cortex, only some of them. We have other steroid hormones produced by other organs like the sex organs, the testes and the ovaries. Now, just to serve as a reminder, here's the adrenal gland. It sits atop of the kidneys. However, the adrenal gland has two parts to it. Here we have an outer shaded part, the outer part shaded in green, and this is the outer cortex. It is the cortex we've just mentioned, which is responsible for the synthesis of corticosteroid. But don't forget that the adrenal gland has an inner part, which is called the medulla. Here we have the inner part shaded in black, and this inner part is called the medulla. And the medulla has nothing to do with the synthesis of corticosteroids. The medulla produces an entirely different class of chemicals called catecholamines. And we can make this information stick in our minds by remembering that cortex ends with X, and we all know that the word X means outer. And so cortex is always the outer part of any organ. Now, do you think that we only have a single type of corticosteroid? No, actually we have three different kinds of corticosteroids. Here we have, the first type of corticosteroid is the mineralocorticoids. The mineralocorticoid function has to do with the regulation and homeostasis of minerals, mainly sodium and potassium. So mineralocorticoid is going to act on the kidneys to reabsorb sodium, and in its place, it's going to secrete from the urine back into the bloodstream, and in its place, it's going to secrete potassium from the blood into the urine. So here's a drawing that would help you make sense of all the information. You see this yellow fluid right here? It is the urine, and as we all know, the urine is lined by kidney cells called renal tubular cells, and besides the renal tubular cells, we have the bloodstream. The red tubes are the bloodstream. Now, the mineralocorticoid is going to diffuse across the cell membrane of the renal tubular cells and bind to intracellular receptors, and it's going to induce modifications on the cell in order to enable the cell to reabsorb more sodium from the urine back into the bloodstream. And in its place, potassium is going to be secreted from the bloodstream into the urine. Now, I'm not going to delve into the importance and the functions of sodium and potassium. That's physiology stuff. My main focus in this video is the biochemistry. I'm going to focus on the biochemical pathway. My goal is to help you make sense and memorize the biochemical pathway. Now, the main mineralocorticoid in our bodies is aldosterone, and this aldosterone is produced by the outer layer of the adrenal cortex called the zona glomerulosa. This drawing shows you the three layers of the adrenal cortex. The outermost layer, colored in green, is the zona glomerulosa. The middle black layer is called the zona fasciculata, whereas the innermost layer, colored in red, is the zona reticularis. And let's not forget that the adrenal gland has a deep part which is the adrenal medulla, represented by the blue X. However, we're going to ignore the adrenal medulla because it has nothing to do with the synthesis of corticosteroids. So we're just going to ignore it. Now, let's talk about the second type of corticosteroids. The second type is called glucocorticoids, and the main glucocorticoid in our body is cortisol. Now, cortisol has that bad reputation of being the stress hormone with a myriad of negative adverse effects. However, cortisol is only bad when in excess. At normal levels, cortisol is very vital to your daily functioning, because had it not been for cortisol, your glucose levels would drop to very dangerous level. Remember, cortisol is a glucocorticoid, and thus it is very important in a glucose homeostasis. Without it, glucose levels would drop and you'd enter a state of hypoglycemia, which can be very dangerous. Now, cortisol is produced by the middle layer of the adrenal cortex called the zona fasciculata. And the last type of the corticosteroids are the sex hormones. Please bear in mind that the main site of sex hormone synthesis is not the adrenal cortex. Actually, it is the sex organs. The sex organs are the main site of sex hormone synthesis, and the majority of sex hormones are produced by the sex organs, the testes and the ovaries. However, the adrenal gland produces a minority of these sex hormones, and the layer that produces the sex hormones is called the zona reticularis. And just to serve as a reminder, we have two types of sex hormones in our bodies. We have the male sex hormones called the androgens, and we can remember it this way: Andrew is a male name, and so androgens are the male sex hormones, whereas the female sex hormones are the estrogens. And in the male, we have different types of androgens. However, the main androgen is testosterone. Whereas in females, the main estrogen is estradiol. And please don't forget that even females have a little bit of testosterone, a little bit of androgens in their bodies, but at very low quantities. And whereas the males, even the males, have very low quantities of the female estrogens. You see? So the predominant sex hormones in the males are androgens, whereas the predominant sex hormones in females are the estrogens. Now, all the corticosteroids, no matter their type, come from cholesterol. Their building block is cholesterol. Cholesterol is going to get converted into pregnenolone via the action of an enzyme called desmolase, and then pregnenolone is going to undergo a series of chemical modifications in order to yield each of the corticosteroids. And so, for example, if pregnenolone wants to yield aldosterone, pregnenolone will undergo a sequence of chemical modifications. However, the sequence of chemical modifications yielding aldosterone is going to be different from the sequence of chemical modifications that would yield cortisol from pregnenolone, and it's going to be different from the chemical modifications that would yield the sex hormones. Keep this in mind. Now, let's talk more about the structure of cholesterol and how it gets converted into pregnenolone. Here's cholesterol. It contains around 27 carbons. That's the total number of carbons present in cholesterol. However, what I care about more is the component of cholesterol, or the components of cholesterol. Cholesterol consists of two major parts. The first part is called the steroid nucleus. The steroid nucleus consists of 4 fused rings, as we can see. Here's A, B, C, and D. And at carbon number 17 of the steroid nucleus, we'll have a hydrocarbon tail attached. The hydrocarbon tail is the second component of the steroid hormones. The hydrocarbon tail is the second component of cholesterol. You see? Now here we have an enzyme called desmolase. This desmolase is going to cut some bonds between the hydrocarbon tails, which is going to act at the bond between carbon number 20 and carbon number 22 of the hydrocarbon tail, and thus desmolase is going to remove a huge chunk of the hydrocarbon tail from cholesterol, and thus cholesterol gets converted into pregnenolone. And notice how that the hydrocarbon tail of pregnenolone is much shorter than that of cholesterol. And we can remember the action of desmolase via this smart hint. Now, we all know about desmosomes, right? You all know about desmosomes, the bonds that link the cells together. Desmolase is gonna break down the bonds present in the hydrocarbon tail in order to remove a portion of the hydrocarbon tail. So the name desmolase actually makes perfect sense. Now, pregnenolone is going to undergo a series of chemical modifications. I'm going to pause the video for a while in order to sketch the chemical modification. So here are the sites of the chemical modifications. Now, the first site of the possible chemical modification is going to be carbon number 17. You see, carbon number 17 sounds attractive. I mean, it serves as a cross-link between the steroid nucleus and the hydrocarbon tail. And so it's very attractive for chemical modifications to occur. And the possible chemical modification that can occur at carbon number 17 is the addition of a hydroxide group. You see, we can add a hydroxide group by an enzyme called 17-hydroxylase. And please note that this step of hydroxylation at carbon 17 doesn't occur in all corticosteroids. Like, for example, if I want to synthesize aldosterone from pregnenolone, then pregnenolone is not going to undergo any hydroxylation at carbon number 17. So the hydroxylation occurs only if I want to synthesize either cortisol or the sex hormones from pregnenolone. Now, the second possible chemical modification is at carbon number 3. You see, carbon number 3 of the steroid nucleus has that huge hydroxide group bouncing right in front of us. I mean, this sounds like an attractive chemical modification too. And actually, the chemical modification that occurs at the hydroxide group of carbon 3 is very essential, and it occurs in all corticosteroids. And so what's the possible chemical modification? I mean, the possible chemical modification, of course, won't be the addition of another hydroxide group. We already have a hydroxide group at carbon number 3. However, we can remove a hydrogen from the hydroxide group via the action of an enzyme called dehydrogenase. You see, its name makes perfect sense. We are removing a hydrogen, dehydrogenase. We're removing a hydrogen from the hydroxide group, and thus the hydroxide group would get converted into a keto group. You see, a double bond is going to form between the carbon number 3 and oxygen because we've just removed a hydrogen. Now, but dehydrogenase is not the full name of the enzyme. The enzyme, before it removes the hydrogen from the hydroxide group, the enzyme acted on a hydroxysteroid. You see, before the removal of the hydrogen group, we had a steroid nucleus along with the hydroxide group, and so the name of the substrate of this enzyme is hydroxysteroid. Here's the hydroxy group and steroid. Here's the steroid. Here's our steroid. And so the full name of the enzyme is going to be 3-beta, 3 because it acts at carbon number 3, 3-beta hydroxysteroid dehydrogenase. And please bear in mind that this step is very essential. It occurs in all the biochemical pathways of corticosteroid synthesis. We can find it right here, we can find it right here, and we can find it right here. Now, I should have wrote the 3-beta hydroxysteroid dehydrogenase with the red color in order to emphasize its importance. However, it doesn't really matter. The color doesn't really matter. Now, what about the rest of the chemical modifications? We have other secondary chemical modifications. What are they? We can hydroxylate carbon number 21 and carbon number 11. You see, via the action of enzymes. I mean, if we want to hydroxylate carbon number 21, we'll have the enzyme called 21-hydroxylase. And if we want to hydroxylate carbon number 11, we'll have 11-beta hydroxylase. And the last possible chemical modification is the removal of the hydrocarbon tail. I mean, this hydrocarbon tail sounds it's more of a nuisance, right? I mean, it's very disturbing. So why not remove the rest of the hydrocarbon tail? And this action is accomplished by an enzyme called lyase. Why is it called lyase? Because it lyses, because it breaks the rest of the hydrocarbon tail. And the full name of the lyase enzyme is 17-20 lyase, because it removes the hydrocarbon tail present between carbon number 17 and carbon number 20. You see, now we are ready to delve into the biochemical pathways. So here's our pregnenolone. We got our pregnenolone from the cholesterol via the action of the enzyme dismolase. Now, the word pregnenolone sounds similar to a pregnant lady, right? I mean, a pregnant lady gives birth, and so pregnenolone is going to give birth to all of these corticosteroids. Now, before we delve into the biochemical pathways, let's be a little bit creative and make up a mnemonic. You see, we all know about the 17-hydroxylation. Okay, and we all know that the 17-hydroxylation only occurs if we want to synthesize glucocorticoids or sex hormones. And so, let's associate the 17-hydroxylation with the age 17. This is a mnemonic. So let's associate it with the age 17. At the age of 17, you're facing the stresses of being a teenager. 17 years old, you're still a teenager, and so you're facing the stresses of being a teenager, in addition to you're still undergoing puberty. And so at 17 years old, your cortisol levels would be high, and your sex hormones would be high. Therefore, using this mnemonic, we can link the 17-hydroxylation with the production of either cortisol or the sex hormones. You see, you can go through either of these biochemical pathways. And of course, the rest of the chemical modifications are going to be different for each of these two corticosteroids, the glucocorticoids and the sex hormones. You see? And so, let's put this mnemonic aside. Let's start with the biochemical pathways. You see? Here's the zona glomerulosa. The zona glomerulosa has its pregnenolone ready, and the zona glomerulosa wants to synthesize mineralocorticoids from the pregnenolone. And so we're going to ask Mr. Pregnenolone a question. Mr. Pregnenolone, you want to synthesize mineralocorticoids. Would you like to undergo 17-hydroxylation? Pregnenolone was going to answer no, because pregnenolone, the zona glomerulosa wants to synthesize aldosterone. There's no need for 17-hydroxylation. And so pregnenolone would not undergo any 17-hydroxylation. And so let's erase. If pregnenolone wants to synthesize mineralocorticoids, no 17-hydroxylation would occur. And so what's the other major modification that can happen? The hydroxide group at carbon number 3. And so the pregnenolone will undergo dehydrogenation at carbon number 3 by the action of the enzyme 3-beta-hydroxysteroid dehydrogenase. And so here's the enzyme, dehydrogenase, and it converted pregnenolone into progesterone. Now, progesterone is going to undergo a series of the secondary chemical modifications. And so the 21-hydroxylation is going to occur at carbon number 21, and an 11-beta-hydroxylation is going to occur at carbon number 11. And remember it this way, always the bigger number precedes the small number. And so 21-hydroxylation is going to precede the 11-beta-hydroxylation. And there's going to be no cutting. The lyase enzyme would have no action on the rest of the hydrocarbon tail. And so here's our progesterone. The progesterone is going to undergo the secondary chemical modifications. 21-hydroxylase will hydroxylate the 21 carbon in order to yield the oxycorticosterone. And then 11-beta-hydroxylase is going to perform another hydroxylation to yield corticosterone. And then corticosterone would get converted into aldosterone via the action of the enzyme aldosterone synthase. Now, you see, progesterone has yielded deoxycorticosterone. This deoxycorticosterone is only one oxygen short of becoming corticosterone, and this oxygen is provided to deoxycorticosterone via the action of 11-beta-hydroxylase. 11-beta-hydroxylase is going to provide a hydroxyl group that has an oxygen atom to the oxycorticosterone, and thus we're covering the lack of oxygen in the oxycorticosterone, and thus the deoxycorticosterone would become a full corticosterone, not a deoxy. It's going to become a full corticosterone, and then via aldosterone synthase, corticosterone gets converted into aldosterone. Now let's talk about the biochemical pathway of glucocorticoid synthesis. Here's the zona fasciculata, and the zona fasciculata has its pregnenolone ready, and it wants to synthesize glucocorticoids from the pregnenolone. And so let's ask Mr. Pregnenolone a question right here. Mr. Pregnenolone, would you like to undergo 17-hydroxylation? Mr. Pregnenolone would answer yes, because remember the mnemonic, I want to synthesize a glucocorticoid, I want to synthesize a stress hormone. And so pregnenolone undergoes 17-hydroxylation in order to form 17-hydroxypregnenolone, and the name makes perfect sense. A hydroxyl group at carbon number 17 by the action of 17-hydroxylase. Then, once we're done with this hydroxylation step, we go through the dehydrogenation step at carbon number 3. And so here comes the enzyme 3-beta-hydroxysteroid dehydrogenase, which would convert 17-hydroxypregnenolone into 17-hydroxyprogesterone. Now, please bear in mind that these two steps, the steps of 17-hydroxylation and the 3-beta dehydrogenation, can be flipped. Pregnenolone, you see this pregnenolone? We can dehydrogenate it first. Here's our pregnenolone. We can start first with the dehydrogenation step and then go across the 17-hydroxylation step. So the matter is flexible. You see, we can start with the 17-hydroxylation or the dehydrogenation whatever it is. However, if we want to start with the dehydrogenation step, pregnenolone would get converted to progesterone, and then progesterone, via the action of the 17-hydroxylation step, it's going to get converted into 17-hydroxyprogesterone. You see, all roads lead to Rome. And so what really matters is that at the end, we have our 17-hydroxyprogesterone ready, no matter the initial steps. Whether you start with the 17 or carbon number 3, here we have our 17-hydroxyprogesterone ready, and then the 17-hydroxyprogesterone would undergo the secondary chemical modifications, which are the hydroxylation at carbon number 21 and carbon number 11. And so 21-hydroxylase is going to convert 17-hydroxyprogesterone into 11-deoxycortisol. You see 11-deoxy, it's lacking an oxygen at carbon number 11, which is provided by the 11-beta-hydroxylase, and then 11-deoxycortisol gets converted into cortisol. And we can remember it this way. You see, if we want to synthesize cortisol, we're gonna provide hydroxylation and not the cutting step of the hydrocarbon tail. You see, if I want to synthesize the glucocorticoids from the 17-hydroxyprogesterone, I'm gonna perform the hydroxylation step instead of the cutting steps. You can go through either of these. I mean, not both of them. You can undergo, or the steroid intermediate can undergo either the hydroxylation step at 21 and 11 or the cutting step. You can't have both, you see? And so cortisol is going to undergo the hydroxylation step instead of the lyase step. And we can remember it this way. In stress response, when cortisol wants to skyrocket at stress response, you want to hide. In stresses, you want to hide. And so this mnemonic would help us associate the hydroxylation at 21 and 11 with cortisol. Now, if I want to synthesize the sex hormones from the pregnenolone, just give me a minute to erase all of this chaos. Now, if I want to synthesize, it's very chaotic. Now, if I want to synthesize the sex hormones from pregnenolone, so we'll start with the major chemical modifications. I mean, we're going to ask Mr. Pregnenolone about the major chemical modifications, which are 17-hydroxylation and the 3-beta dehydrogenation. And so Mr. Pregnenolone is going to get asked, Mr. Pregnenolone, you want to synthesize sex hormones. Would you like to undergo 17-hydroxylation? He's going to answer yes. And so here we have our 17-hydroxypregnenolone ready. However, we're going to ask Mr. Pregnenolone or 17-hydroxypregnenolone whether it wants to undergo the lyase step. And of course, it's want to undergo the lyase step because it wants to synthesize the sex hormones. And so the 17-hydroxypregnenolone is going to undergo the lyase step. We're going to remove the rest of the hydrocarbon tail in order to yield what? In order to yield dihydroepiandrosterone. You see, this steroid intermediate fits its name. It's an androsterone. It's a sex hormone, andro, from androgen. So dihydroepiandrosterone is a weak androgen. And this dihydroepiandrosterone can undergo a series of chemical modifications in order to yield other sex hormones. You see? And so pregnenolone was hydroxylated at carbon number 17 and had its hydrocarbon tail ready. However, we still need to dehydrogenase the hydroxide at carbon number 3. This is a very essential step. Please don't forget it. This dehydrogenation step has to occur in all corticosteroids. And so the dihydroepiandrosterone can undergo 3-beta hydroxysteroid dehydrogenation to yield androstenedione. You see, and please bear in mind that this matter is flexible. I mean, pregnenolone. You see this 17-hydroxypregnenolone? The 17-hydroxypregnenolone can undergo the dehydrogenation step before the lyase step. When 17-hydroxypregnenolone undergo the 3-beta hydroxysteroid dehydrogenation step, it's going to get converted into 17-hydroxyprogesterone, and then by the action of 17-20 lyase, the 17-hydroxyprogesterone would yield androstenedione. You see, the matter is flexible. And there's a third detour that pregnenolone can take in order to reach androstenedione. So we can start with the dehydrogenation step first. We can dehydrogenase the pregnenolone to yield progesterone, and then progesterone is going to get 17-hydroxylated to yield 17-hydroxyprogesterone, and then after that, the lyase would show up and convert 17-hydroxyprogesterone to androstenedione. Please pay attention to this. Have you noticed that we can't undergo the lyase step before the 17-hydroxylation step? You see, that's a pattern. You can't perform, the lyase enzyme can't function without hydroxylating the 17 carbon first. Why? Because only when you hydroxylate the 17 carbon would the rest of the hydrocarbon tail be unstable enough for the lyase enzyme to separate it and remove it from the steroid nucleus. You see? So the lyase step can't precede the 17 hydroxylation step. You see, so what really matters is that at the end we have the androstenedione. Then this androstenedione is going to convert it into either testosterone or estradiol via the action of other enzymes. And notice something. The sex hormone, the androstenedione, had its lyase step done. So we can't perform the hydroxylation step at carbon number 21 and 11. Once again, you can undergo either of these secondary modifications, which is hydroxylation or the lyase, but not both. And so, since androstenedione has undergone the lyase step, we can't perform any hydroxylation on it. And so, we won't bring up any hydroxylase enzymes. We're going to bring up a whole different bunch of enzymes. Like if I want to synthesize testosterone from androstenedione, I'm going to bring an enzyme called 17-ketoreductase, and this 17-ketoreductase is going to convert androstenedione into testosterone. Now, if the person was a male, a very small fraction of testosterone might get converted into estradiol via the action of an enzyme called aromatase. You see, so aromatase converts the male androgen, the main male androgen, which is testosterone, into estradiol, which is the female sex hormone, the main estrogen in females. However, in males, only a small fraction gets converted. But in females, a huge fraction of testosterone gets converted into estradiol. And we can remember this step via this way. You see, aromatase. The word aroma in English means sweet smell, like something that smells similar to perfumes. And it's a soft smell. And so you can link this with testosterone becoming softer and yielding the female sex hormone. Now, another step that androstenedione can undergo is to actually flip these enzymes and to begin with the aromatase first instead of beginning with 17-ketoreductase. And so, remember that aromatase would convert the androgens into estrogens. You see? Aroma, soft, sweet, perfume-like smell, and so the androgen becomes softer. And so aromatase would get converted, aromatase would convert androstenedione into estrone. Estrone is a type of female estrogens. You see, estrone is a type of female estrogens, and then 17-ketoreductase would convert the estrone into estradiol. Now, that's it for the lecture. I hope you guys enjoyed it. I hope I've made things simpler for you, that you've memorized a huge chunk of this lecture. Please subscribe to my Instagram channel, subscribe to our Telegram channel, and whatever question you have in mind, don't hesitate to contact me. Thank you all and good luck.